So what is a pipette? A pipette is just a device that's used to transfer small microliter amounts of liquid. Well, how big is a microliter? 1,000 microliters is equivalent to 1 milliliter. 1,000 milliliters is equivalent to a liter. So there are actually 1 million microliters in 1 liter. So let's make this a little bit more visual. You have a 2-liter Diet Coke, that's 2 million microliters. Additionally, 10 microliters is approximately the size of a small raindrop, not the huge ones that we get here during hurricane season in Louisiana. And our pipettes in our classroom are designed to hold 1,000 microliters or less. So this would be the equivalent of 1 milliliter of liquid. So let's choose a pipette. You'll notice that your table should have 4 different pipettes. This is a P10, a P20, a P200, and a P1000. These names correspond to the maximum amount the pipette can hold. So a P10 can hold up to 10 microliters, while a P1000 can hold up to 1,000 microliters. The maximum volume of a pipette can be listed either on the top of the pipette or on the side. Look at the pipettes at your workstation or check the ones out on the slide. Can you find the one that holds up to 20 microliters? What about 200? Many pipettes also have a lower range printed on the top or side. If you need more than the maximum that that pipette can hold, go up a size. If you need less than the minimum, go down. So let's look at this set of 4 pipettes. You can see that the minimum to maximum range is printed on the side. Which of these pipettes should we use to transfer 250 microliters? How about 15 microliters? Let's try 1. $20 microliters? Some of you guys may be saying this one. Others of you may be thinking this guy. The truth is you can actually use either of these 2 pipettes to transfer 20 microliters. Last, how about 1,700 microliters? We could use this guy, the P1000, but we have to use it twice. It's really important that we never adjust the pipette above the maximum volume that it's set to go. So how do we set the volume? When we set the volume, we'll turn the dial at the top of the pipette. So that can be either the white dial at the top or the black dial a little bit further down. Either of these should work. To know what volume we are currently set at, we look at the volume setting window. So how do we read this? To understand how to read pipettes, let's take a look at the next slide, noting that the volume setting window has 3 places. So depicted here are the volume setting windows for the P10, P20, the P200, and the P1000. So this may be a little bit confusing at first, but a good rule of thumb is that the top number in the window corresponds to the numeric place of the maximum volume that that pipette can hold. For example, let's look at the P10 and the P20. The P10 can hold 10 microliters. The P20 can hold up to 20. 10 and 20 are both in the tens place, making the second window the ones place and the third window a decimal. In the P200, we can hold up to 200 microliters, making the top window the hundreds place, the middle window the tens place, and the bottom window the ones place. So how is the P1000 laid out? Take a moment and see if you can figure it out. You should hopefully figure out that the top number belongs to the thousands place, The middle number is the hundreds place, and the bottom number is the tens place. Let's practice this a little bit. On the previous slide, we note that we all have the same volumes. The reading will be different depending on which pipette we have. These all appear to be set at some version of 73, but you can see that for both the P10 and the P20, we're looking at 7.3 microliters. For the P200, this is the hundreds place, there aren't any. Tens place, ones place, this should be 73 microliters. Looking at the biggest, the P1000, remembering that the top is going to be in the thousands place, the middle in the hundreds place, and the bottom in the tens place, we arrive at 730 microliters. On this slide, at what volume is each pipette set? The P10 should be 10 microliters. The P20, also 10 microliters. The P200, 100 microliters, and the P1000, 1000 microliters. Let's take a look at the next slide. So something's wrong here. See if you can pinpoint what it is. Hopefully you notice that the first P10 is set at 17 microliters. That's higher than the maximum volume. Additionally, the P1000 is set higher than it can go, at 1,700 microliters. The other two volumes are acceptable. It's very important that we don't go over the maximum volume when setting the pipette, as this can damage the internal mechanism. So now that we've set our volumes, let's go ahead and choose tips. You should always use a tip on the pipettes. This can prevent damage to the pipette and contamination between samples. So one of our goals from pipetting is never to allow liquid to go up into the barrel or small hole at the bottom of the pipette. Tips will help prevent that. You'll notice that there are three sizes of pipette tips at your table. Small, which are kind of orange, medium in a yellow tray, and large in a blue tray. If you choose the wrong tip, you risk damaging the pipette or being unable to draw up liquid. The small tips always go with the P10 only. The medium tips are for both the P20 and the P200. The large tips are only for the P2000. So let's look at an example of getting tips and how to tell if you've gotten the wrong size tip on your pipette. Now that we know a little bit more about choosing tips, let's go ahead and talk about how to take up samples. So to take up samples, you'll hold the pipette vertically and using your thumb should depress the plunger in a controlled way. About 1/2 to 3/4 of the way down, you will feel sort of a natural stop. You can see this depicted in the second image here. This is called the first stop. If you push a little harder, the plunger will go down all the way. This is called the second stop. To take up liquid, first depress the plunger only to the first stop before immersing your tip. Going to the second stop at this point will cause you to pull up a completely inaccurate volume. Place your tip pipette into the liquid and using a very controlled motion, allow the plunger to return to its starting position. It's very important that you don't just allow the plunger to snap back uncontrolled because this may cause liquid to go up into the barrel of the pipette. Now that we have fluid in our tips, we're ready to expel our sample into a new tube. In order to do so, place the tip of your pipette into the bottom of a clean tube and depress the plunger all the way down, this time to the second stop. Pressing down to the second stop will now ensure that all the liquid in the pipette is expelled. You can now eject the used tip from the pipette into a waste beaker by pressing the gray button that's just beside the plunger. We should always remember to change tips between any samples that we're pipetting.